This method can help to produce large amounts of recombinant RNAs of interest for application in research or industry. The main advantage is that recombinant RNA is produced in Escherichia coli in a cheap process that can be easily scaled up. Importantly, the RNA is produced on a circular RNA scaffold, which facilitates purification to homogeneity. In contrast to DNA or proteins, RNAs of interest are not easily produced in large amounts in biofactory systems, such as Escherichia coli culture. Our method is based on co-expression of the RNA of interest inserted into a highly stable circular scaffold and a plant ligase that mediates RNA circularization. The circular RNA scaffold derives from eggplant latent viroid. Viroids are relatively small, non-coding, highly base-paired circular RNAs that are infectious to some higher plants.
We can produce tens of milligrams of the recombinant RNA per liter of bacteria culture in regular laboratory conditions.
To begin this procedure, amplify the cDNA by PCR as outlined in the text protocol. Next, add 100 nanograms of the plasmid PLELVD-BZB to a 0.5 milliliter tube. Add 10 U of the type 2S restriction enzyme BPI1 and enough of buffer G to create a 20 microliter reaction. Incubate at 37 degrees Celsius for 1 hour to digest the plasmid. After this, separate the PCR and digestion products by electrophoresis in a 1% agarose gel in TAE buffer.